Assalamualaikum, my name is Balqish Nadhirah Binti Sahlan, and today I will be presenting my final year project on the antimicrobial activity of lactic acid bacteria isolated from fermented durian flesh against pathogenic bacteria. Now we will move to the introduction. Tempoyak or fermented durian pulp is known to contain lactic acid bacteria, which is also known as LAB, which have the potential to act as probiotics and natural antimicrobial agents. However, antimicrobial resistance has become a major global health issue. Many pathogenic bacteria are developing resistance due to frequent and improper antibiotic use. This research aims to explore the antimicrobial potential of LAB against S. aureus and E. coli to provide insights into natural alternatives to antibiotics. Next, the objectives of this study are to determine the total number of lactic acid bacteria present in 1-week-old tempoyak sample, then to study the biochemical characteristic of lactic acid bacteria isolated from tempoyak, and to evaluate the antimicrobial activity of lactic acid bacteria against S. aureus and E. coli. Now we will move to the literature review. LAB exhibit two primary mechanisms of antimicrobial activity, which is the production of organic acids such as lactic and acetic acids, lowers pH and creates an unfavorable environment for pathogens. Additionally, the generation of hydrogen peroxide damage bacterial cells. These effects make LAB against both gram-positive and gram-negative bacteria are more effective. LAB also convert carbohydrates into lactic acid, producing ATP in the process. This acidification contributes to its antimicrobial properties. LAB can be classified into two groups. First is homofermenters, which produce mostly lactic acid from glucose, and heterofermenters, which produce lactic acid and carbon dioxide, acetic acid, and ethanol. Given these properties, LAB and their metabolites can be considered a natural alternative to antibiotics. LAB are widely used in various fields. In the food industry, fermentation helps preserve food, enrich vitamins, and inhibits pathogens. In the medical field, LAB produce bacteriocins and organic acids, which can serve as an alternative treatment to combat antimicrobial resistance. In agriculture, LAB supports sustainable farming, detoxify chemicals, and promote soil fertility. Okay, now we will move to the methodology. To isolate LAB from the sample, 1 gram of tempoyak was mixed with 10 ml of 0.85% NaCl. The mixture was serially diluted up to 10 to the power of -6. Next, 0.1 ml of each dilution were evenly spread on MRS agar, and each dilution were made duplicate. Next, after the incubation for 24 hours at 37 degrees Celsius, the plates that had colonies between 300 and 30 was chosen. Then, the average colonies of the selected plates were calculated. The colony forming unit per milliliter, which is CFU per ml, of the selected plates were also calculated. This method aligns with objective 1. Now, 48 colonies were picked and transferred onto MRS agar that contained cyclohexamide. Then, the plates were incubated for 24 to 48 hours at 37 degrees Celsius. Then, the colonies that grew on the agar were observed. Now, for the biochemical tests of LAB, 5 steps of biochemical tests were done to evaluate the biochemical characteristic of the selected LAB. The first test is gram staining, second test is catalase test, third test is carbohydrate fermentation test, which tests 3 sugars, which are lactose, glucose, and sucrose. The fourth test is NRVP, and the last test is SIM test. Now we move to the antimicrobial assay, which is this division. There are 2 types of LAB tested in this division. The first test is direct LAB, the second test is neutralized LAB. Okay, firstly, the LAB inoculum was prepared, and overnight culture of S. aureus and E. coli also was prepared. Then, for the direct LAB, the LAB inoculum was centrifuged at 4000 rpm for 10 minutes. Then, 50 microliter of the supernatant was transferred into the sterile disk. The bacterial lawn of S. aureus and E. coli were done on MH agar. Each paper disc that containing LAB supernatant were transferred onto the agar. Then, the plates were incubated for 24 hours at 37 degrees Celsius. After the incubation, the zone of inhibition were observed and measured. For the neutralized LAB, the pH of the supernatant of the centrifuge LAB inoculum was checked by using the pH paper. Then the supernatant were neutralized with NaOH until the pH turned 7. Then, after the neutralization, 50 microliter of the neutralized supernatant was transferred onto the sterile disc. Then, each paper disc that containing neutralized LAB supernatant were transferred onto the agar with the respective bacterial load. Then, the plates were incubated for 24 hours at 37 degrees Celsius. After the incubation, the zone of inhibition were observed and measured. Now, we will move to the results and discussion. For the total number of lactic acid bacteria from a 1-week-old tempoyak, the plate 10 to the power of -3 was chosen because the colonies that grew were between 300 and 30. Then the CFU per ml of the plates were counted by using the formula that were given. Now, to isolate the LAB, 48 colonies were picked and transferred onto the MRS agar that contained cyclohexamide. Cyclohexamide was used because in tempoyak sample, fungi and yeast can grow on MRS agar, competing with the LAB. Therefore, cyclohexamide can act as an antifungal agent that inhibits eukaryotic protein synthesis but does not affect bacteria. So, the results showed that there were a total of 9 colonies of LAB that grow on the MRS agar with the cyclohexamide. The biochemical test results were consistent with the Bansal et al. 2013. All LAB strains were gram-positive and catalase negative. The carbohydrate fermentation and MRVP tests matched the reference study, and the SIM test results confirmed no sulfide, indole, or motility activity. Next, for the antimicrobial assay, which is this diffusion result, the result shows that only the positive control, which are the antibiotic discs, which are used here is chloramphenicol, had zone of inhibition against S. aureus and E. coli. Also, from the result that were obtained, all of the LAB strains for both direct and neutralized does not show any inhibition zones against S. aureus and E. coli. Compared to the study of Nadhirah et al. 2019, there were significant zones of inhibition for the LAB strains against S. aureus and E. coli. This might be caused that the method that I use is different from this study, which I use this diffusion, and this study used agar well diffusion. Also, the factors that might affecting the zone of inhibition of the LAB strains also include the differences in LAB strain use, then culture conditions, and next is supernatant concentration, and lastly might be the pH differences. In summary, LAB from tempoyak was successfully isolated and identified their biochemical properties. However, no antimicrobial activities were observed against S. aureus and E. coli. For future studies, I recommend using different antimicrobial assays such as agar well diffusion, growth microdilution, or time kill assays. Expanding the study to include more bacterial pathogens will help assess LAB's broad spectrum potential. That concludes my presentation. Thank you for your time and attention.